Hello everyone, my name is Yoli, and for my IRB presentation, I'll be talking about vitamin B12 deficiency and Crohn's disease. The objective of this study is to examine the efficacy of nasal hydroxycobalamin in the treatment of vitamin B12 deficiency in Crohn's disease in adolescents between the ages of 13 to 19. And to also eventually investigate the possibility of long-term use of intranasal hydroxycobalamin as a maintenance therapy in patients who need vitamin B12 parenterally. I'm going to talk about vitamin B12. What is vitamin B12? Vitamin B12, also known as cobalamin, is a water-soluble vitamin that mainly exists in three forms. The first one being hydroxycobalamin, methylcobalamin, and adenosylcobalamin. Hydroxycobalamin takes effect in the blood and the cell plasma. Methylcobalamin works in the cell plasma, while adenosylcobalamin functions in the mitochondria. Sources of vitamin B12. Vitamin B12 is found mainly in animal products such as liver, beef, lamb, mussels, clams, sardines, dairy products, eggs, and cereals that are fortified with vitamin B12. How is vitamin B12 absorbed in our bodies? Vitamin B12 is absorbed in the terminal ileum. This absorption is almost entirely dependent upon the intrinsic factor, which is a glycoprotein that's secreted by parietal cells that are located in the mucosa of the stomach. The intrinsic factor binds to vitamin B12 from food, and the complex is transported across the cell membrane that is bound to another glycoprotein that's called transcobalamin. What are the functions of vitamin B12? There are several functions of vitamin B12. The first being, it's important in the synthesis of DNA, which influences cell division and blood formation. It's also responsible for energy metabolism, which is vital for energy production in the mitochondria. It's also important in lipid metabolism, for building and maintenance of the cell membrane and the myelin sheath, which is a protector of the nerves in the central nervous system and brain. It's also involved in the synthesis of hormones and neurotransmitters, which are important in the production of crucial neurotransmitters. It's also important in detoxification, which is an important opponent of nitrosative stress. What causes vitamin B12 deficiency? It could be caused by autoimmune pernicious anemia, gastrectomy, ileal resection, pancreatic insufficiency, and malabsorption syndromes, including Crohn's disease and celiac disease. These are inflammatory bowel diseases. And for our study, we're going to be concentrating on the deficiency of vitamin B12 in Crohn's disease. What is Crohn's disease? Crohn's disease is a chronic inflammatory bowel disease which affects the end of the small intestine, also known as the distal ileum. It affects the gastrointestinal tract, mainly the ileum, colon, or both. Inflammation can be in one area and then spread all the way through the intestinal wall. So now we're going to talk about the study of vitamin B12 deficiency in Crohn's disease. For this study, we're going to recruit 9 patients from an outpatient clinic of the Children's Hospital of Eastern Ontario. And these children are adolescents between the ages of 13 and 19. And for this study, we're going to be administering nasal hydroxobalamine containing 700 milligrams of the hydroxobalamine. And it's going to be a preserved solution that will be administered using a metered dose spray device. And for the study, we'll only have patients who have been diagnosed with vitamin B12 deficiency and who also have Crohn's disease and recurring inflammatory activity of the terminal ileum. And then all the patients should have an initial plasma cobalamin level of less than 200 nanograms per liter. This level defines vitamin B12 deficiency. And during the study period, all patients with Crohn's disease should be in remission. Also, patients who have had prior nasal surgery, nasal disease, or who have used transnasal medication before will be excluded from the study. For this study, we'll have the patients, they'll be administered a dose of 1,500 milligrams of hydroxobalamine intranasally, and each nostril will get 70 mils of the vitamin. And this will be administered at day 0, day 7, day 14, day 21, and day 28. The plasma cobalamin concentrations, they will be measured using radioimmunoassay at day 0, 7, 14, 21, and 28 after the first intranasal administration. Then, after each application, the cobalamin levels will be assessed at 1 hour in all the patients. Then the normal plasma levels will be defined between 200 and 700 nanograms per liter. Plasma cobalamin concentrations will be accurately determined up to 2,400 nanograms per liter, and all values greater than that value will be presented as 2,400 nanograms per liter. So from this study, our results indicate that after a single intranasal dose, there's a sustained increase of cobalamin concentration 1 week after application in all the patients. 1 dose of intranasal hydroxobalamine shows that there's an 8-fold increase of mean baseline cobalamin concentration after 1 hour of application. This indicates that there is a rapid absorption of a significant amount of the hydroxycobalamin in the B12 deficient patients. And this you can see in this table that we have. We have patient 1, 2, up to 9. We'll talk about the first patient here. At day 0, which is before the administration of 1,500 micrograms of hydroxobalamine, we have this reading, which reads just a little less than 200, which is, let's say, maybe 199. And then after an administration, it goes up. And then on day 7, there's an increase. Not so much of an increase on day 14, but then on day 21 and 28, we do see a significant increase. And this seems to be the case with the other patients. Patient 2 didn't show much increase in the concentration of plasma cobalamin. So in figure 1 that I just showed you, we can see that there is an increase of baseline cobalamin concentration before and 1 hour after nasal application of hydroxobalamine at day 0, 7, 14, 21, and 28. And then after 1 hour of intranasal hydroxobalamine application, there's a large increase in the concentration of cobalamin for all patients. Then the mean baseline cobalamin concentration before hydroxobalamine application on days 0, 7, 14, 21, and 28 was 199 nanograms per liter. And then the mean cobalamin concentration after 1 hour, it increased to 1,730 nanograms per liter. And this indicates an 8-fold increase of the mean baseline cobalamin concentrations. So from these results, we can tell that 1 dose of nasal hydroxobalamine will lead to an 8-fold increase of mean baseline cobalamin concentration 1 hour after being administered. The data suggests that a weekly intranasal administration of 1,500 milligrams of hydroxobalamine will be enough to restore vitamin B12 deficiency. And the sustained increase in baseline cobalamin concentration 1 week after nasal application of hydroxobalamine will suggest that the hydroxocobalamin has been absorbed in the body. Some studies suggest that hydroxobalamine may be the better form of vitamin B12 for treating the deficiency, because this form of vitamin B12 lacks the cyanide moiety and has a hydroxyl group instead, and this makes it more efficient. And lastly, I have my references. Thank you for listening.